Welcome to Meiosis in Motion, brought to you by McKaela and Kristen. Mitosis and meiosis are two types of cell division that are important for allowing organisms to pass on DNA and reproduce. In this video, we will be focusing on animal cells going through meiosis and not plant cells because both cells have a slightly different process. The main difference between mitosis and meiosis is that mitosis results in two genetically identical diploid cells. Diploid means that each cell has two chromosomes at the end of mitosis. N equals 2, which is the number of chromosomes. At the end of mitosis, 2n equals 4, so each cell has 4 chromosomes. In meiosis, 4 genetically different haploid cells are created. Haploid means that the cells have half the number of chromosomes as the parent cell. N equals 2, so at the end of meiosis, each cell has 2 chromosomes. Each cell that is created from meiosis is genetically different, which helps organisms sexually reproduce and have genetic variation between their offspring. Now let's get into the exact steps of meiosis in depth.
The step before meiosis officially begins is interphase. During interphase, which is also the longest phase, the cell grows, duplicates DNA, and prepares for cell division. The DNA is in the form of chromatin in interphase. The second step is prophase 1, where the chromatin starts to condense into chromosomes and become visible. DNA crosses over from one chromosome to the other, producing genetically different chromosomes, and the nuclear envelope starts to break down. Additionally, the nucleolus disappears, and the centrioles move to the opposite ends of the cell. Next is metaphase 1, and in this step, the pairs of chromosomes line up in the middle of the cell. Next, in anaphase 1, the spindle fibers pull apart the pairs of chromosomes to the cell's poles. The cell then moves to telophase 1, where the cell pinches in the middle and a cleavage furrow develops. At the end of telophase 1, cytokinesis occurs and 2 daughter cells are created. The sixth step is prophase 2, and the chromatin condenses into chromosomes and the nuclear envelopes of both cells break down. In prophase 2, the crossing over chromosomes does not occur, and the chromosomes cannot be seen. In metaphase 2, single chromosomes line up in the middle of the cells. The eighth step is anaphase 2, and the spindle fibers pull apart the chromosomes and the sister chromatids to opposite ends of the cells. The last step of meiosis is telophase 2, where both cells pinch in the middle and a cleavage furrow is formed. Cytokinesis also occurs in both cells, and 4 unique daughter cells are formed. After reviewing all the steps of meiosis, let's see the entire process one more time. To recap, cells that divide via meiosis go through prophase, metaphase, anaphase, and telophase twice, producing 4 haploid daughter cells that are genetically different from the parent cells because of the process of crossing over of chromosomes that occurs in prophase 1.
Thanks for watching. We hope you learned a little bit more about meiosis.